Hey everyone, my name is Paulina, and today I will present a review of a scientific paper that talks about how the health of our microbiome is associated with the way we live, what we eat, um, and diseases that can emerge from an unhealthy lifestyle and medication intake. So, the gut microbiome's role in health and disease has been extensively studied, with associations found between specific microbes and various disease states. This research paper explores the relationship between the gut microbiome and cardiometabolic traits, and the hypothesis is that the gut microbiome composition is associated with cardiometabolic traits. And researchers aim to explore the relationships between the gut microbiome composition and diversity, and how these factors relate to lifestyle, diet, clinical phenotypes, blood markers, and medication intake. The research in this paper uncovers microbial factors that can impact cardiometabolic traits by controlling host inflammation and metabolism. And by identifying microbial taxa associated with cardiovascular risk, diabetes, and metabolic syndrome, the study aims to clarify the molecular mechanisms through which the microbiome impacts health and disease. The question posed in this research was how the gut microbiome associates with cardiometabolic traits, and the authors employed 16S rRNA gene sequencing to analyze the microbial composition and its relationship to clinical phenotypes, medications, and blood markers. So the bottom line up front is that research in the microbiome field reveals the impact of gut microbes on health, diseases, and treatments, shedding light on conditions like cardiovascular disease, diabetes, and obesity. Lifestyle factors like diet and exercise significantly influence microbial diversity, and specific microbial species like Ruminococci clostridialis, Lactospira C. bacteroidetes, and firmicutes are linked to prevalent CVD, diabetes, and various clinical traits. So now let me explain the experimental design. The study conducted 16S rRNA gene sequencing on stool samples of 423 participants, and the data processing involved the use of UParse pipeline and 16S biobakery workflow. The UParse pipeline is a bioinformatics tool used for processing 16S rRNA by performing quality control to help analyze and interpret microbial data. And 16S rRNA biobakery is a computational analysis pipeline designed for processing, analyzing, and interpreting microbiome sequencing data. UParse and 16S biobakery collaborate in a complex process to achieve optimal results. So now I will briefly summarize the methods used to conduct the research. The Framingham Heart Study conducted microbiome analysis on 3 cohorts, Generation 3, Omni 2, and New Offspring Spouse. They used 1,702 stool samples from the 1,423 participants, along with 279 technical replicates to evaluate processing variability. Since most participants use medications, they classified them into 4 categories: antihypertensive agents, insulin and hypoglycemic medications, LDL lowering, and triglyceride lowering medications. They also considered the current diabetes status and calculated a 10-year cardiovascular disease risk on participants who did not have any cardiovascular disease diagnosis. Lifestyle factors like diet and physical activity were also included. So here is figure one, and it just shows the summary of the data types and demographics. It sets the stage for understanding the context in which the microbial association analyses were conducted. You can see here under letter A, it shows the different data types that were collected, like the diet and stool samples. And under letter B, it shows histograms of demographics of the cohort showing various demographic factors like age, sex, BMI, race, metabolic syndrome concerning BMI distribution, CVD risk, and disease prevalence. This part of the figure provides a visual representation of the distribution of these demographic variables within the study cohort. And letter C shows distribution of CVD and diabetes in the cohort with BMI distributions per diagnostic category. So now onto the results. The study revealed that consuming fish, vegetables, fruits, nuts, and cereals within the last week showed the strongest association with diversity in the gut. Factors with trending significance like leisure time exercise, HDL cholesterol, and starch consumption were also associated with microbial diversity, while intake of soft drinks, processed meat, and dairy showed a declining trend in diversity. And as you can see in right here in letter J, down here, the plot illustrates how each variable related to dietary factors correlates with the Shannon diversity, which is a measure of species richness and evenness in the microbial community. And the higher the percent variance, the stronger the association between the specific food item and category. For example, if a dietary factor like vegetables has a high percent variance, it suggests a positive significant impact of vegetable consumption on the microbial diversity within the gut microbiome. And if you look at the triglycerides down here, they are in the negative percent variance. So they imply that high levels of triglycerides in the blood are linked to decreased microbial diversity. There is also another article that I read called Diet Rapidly and Reproducibly Changes the Gut Microbiome. And this study suggests that dietary changes can quickly affect the gut microbiome, which aligns with the current study's emphasis on the association between diet and microbiome diversity. And both studies highlight the importance of diet in shaping the gut microbiome and its diverse composition. And the article went into how disease risk and the gut microbiome are interconnected. And it discovered notable links between specific microbial species and various diagnostic conditions, medications, and blood test results. By analyzing the data, the researchers were able to identify microbial taxa that showed associations with different health conditions and treatments. These associations were observed across a range of diseases, medications, and clinical measurements, providing a comprehensive view of how the microbiome interacts with our health status. If you look at letter A, the histogram shows alpha diversity, which is a measure of species richness within a single sample. And the richness of each sample is presented as the number of operational taxonomic units, or OTUs, per sample. Each bar in the histogram represents a sample, and the height of the bar indicates the number of OTUs detected in that sample. By looking at this histogram, researchers can observe the variation in species richness across samples included in the study. I found another study that talks about an anti-diabetic drug called metmorphin that is able to increase specific beneficial bacteria like Akkermansia and short chain fatty acid producing microbiota. Associations were observed between microbial composition and participant phenotypes like BMI, blood analyte measurements, diagnostic conditions, and medication use. Specific microbial taxa were associated with normal weight and obesity in participants. A detailed analysis pointed out that OTUs showed a connection with the weight status of participants. For example, two OTUs, Lecnospiraceae and Coprococcus, were more abundant in people classified as having normal weight, and in contrast, four OTUs, including two Lecnospiraceae, Ruminococci, and Clostridium, were enriched in participants classified as obese. This distinction in microbial abundance between normal weight and obese participants suggests a potential association between these microorganisms and weight status. If you look at letter I, the x-axis and y-axis represent the non-metric multidimensional scaling, or NMDS, analysis of Bray-Curtis distances calculated from OTU level relative abundances. NMDS is a technique used for visualizing the similarity or dissimilarity between samples based on the microbial composition. The circles in the plot represent individual samples from the study, and each circle is colored according to the Shannon diversity of the sample. The colors indicate different levels of Shannon diversity, helping to visualize how diversity varies across samples. And the arrows in the plot represent the variables that significantly correlate with the ordination configuration, and the length of the arrow indicates the strength of the correlation, and the direction shows the variable that changes most rapidly and with the maximum correlation with the ordination configuration. This helps to identify which factors or variables have the most influence on the microbial composition of the samples. So the relative abundance of bacteroidetes ranged from 0 to 98%, while firmicutes ranged from 2 to 99%. And Shannon diversity index was strongly correlated with firmicutes and negatively correlated with bacteroidetes. Bray-Curtis distance analysis, which is a tool to compare the microbiome compositions of different people based on the presence and abundance of different microbial taxa, showed that the proportion of bacteroidetes and firmicutes was the largest driver of variation across people at the phylum level. If you look at letters B and C, each dot represents a person and is colored by the proportion of bacteroidetes or firmicutes abundance. And in letter B, each dot represents a person's gut microbiome composition. And the closer the dots are, the more similar their microbial compositions are based on Bray-Curtis distances. And the color of the dots indicates the proportion of bacteroidetes in the microbiome. Letter C shows the same thing, but how people cluster based on their firmicutes composition. So the study identified microbial associations with diabetes status and found that all 8 microbes associated with a diabetes diagnosis were significantly associated with insulin and hypoglycemic drugs. This suggests that drug intake may influence the diabetes signal and that the microbiome interacts with these pharmaceuticals. And the study also found that the insulin and hypoglycemic drugs varied Drugs variable showed stronger statistical significance and identified additional microbial associations compared to the diabetes diagnosis alone. Microbial families like Ruminococci, Clostridialis, and Lectospiraceae were found to be significantly associated with cardiovascular disease, diabetes, metabolic syndrome, as well as medications like lipid-lowering, hypoglycemic, and antihypertensive agents. These microbial families also correlated with cardiometabolic phenotypes like body mass index, triglycerides, and glucose levels. This suggests that these specific microbial families are linked to key health parameters related to cardiovascular risk and metabolic syndrome, highlighting their potential role in influencing these health conditions through the gut microbiome. So, like I mentioned before, Lectospiraceae and Coprococcus were increased among people of normal weight, while two Lectospiraceae, Ruminococci, and Clostridium were enriched in obese participants. This specific finding indicates the differences in microbial abundances in participants with different BMI classifications. If you look at letter H, the significance of the relationship between BMI and microbial diversity is shown. It provides context on specific microbial taxa associated with different BMI groups and visually presents how Shannon diversity varies across different BMI classifications, possibly showing a decrease in diversity with an increasing BMI status. This supports the idea that microbial diversity is influenced by body weight, as indicated by specific OTUs, enriched in normal weight versus obese participants. So the article has made significant contributions to the field by linking the gut microbiome to various cardiometabolic traits through a large-scale population-based study. Some key findings include the association between microbial diversity and cardiovascular disease risk, the impact of lifestyle factors like diet and exercise on microbial diversity, and the microbial taxa associated with CVD and type 2 diabetes. These findings provide new insights into the role of the gut microbiome in cardiometabolic diseases. One important takeaway is the identification of microbial taxa that overlap in their associations with different cardiometabolic traits, suggesting shared microbial mechanisms underlying these diseases. The study also highlights the potential for future research to explore how microbiome heterogeneity influences responses to dietary and lifestyle factors in cardiometabolic disorders. The strengths of this study lie in its large sample size, comprehensive clinical phenotyping, and integration of microbiome data with various clinical traits. However, some limitations include the cross-sectional nature of the study, which limits causal inference, and the need for further research to validate the associations observed. Future directions could involve longitudinal studies to track changes in the microbiome over time in people with increased CVD risk, more detailed metagenomic sequencing, integration of host genetic data, and exploration of the impact of microbial factors on disease pathogenesis. So to conclude, the study emphasizes the significance of the gut microbiome as a factor in cardiometabolic diseases, potentially shedding light on the mechanisms behind disease development and progression, and the identified links between microbial diversity, dietary habits, and lifestyle choices highlight the microbiome's role in influencing host physiology and immune responses. These results have implications for personalized medicine, indicating that modifying the gut microbiome's composition and function could provide new strategies for preventing and treating cardiometabolic disorders. By identifying microbial associations with disease risk factors and medications, the study enhances our understanding of the gut microbiome's impact on human health and disease. So here I included the 5 questions that we can discuss in Pursual. And here are the article references for the articles that I mentioned while presenting. And that's it. Thank you so much.